Listen to part of a lecture in a botany class.
Okay, let's move on to bacteria and viruses that can infect plants. And let's start out with a virus that's rather common in various plants, but first became known in connection with tulips. This virus can cause a change in pigmentation that dramatically affects the color of the plant's leaves or flower petals. But since not all cells of the plant tissue are infected, the result tends to be color variation, with color intensified in one part of the flower petal and faded in another. This is called color breaking, and the virus that causes this in tulips is called the tulip breaking virus. The tulip breaking virus is now known to have detrimental effects on plants. They're weaker and sometimes reduced in size. But for centuries, people didn't have a clue about this virus. Not until the early 1900s was it known what caused the color breaking in tulips, what made the tulip plant produce flower petals so radically different in color or in pattern from what you would have expected. Tulip flowers with stripes or streaks or feather or flame patterns on their petals. There's no doubt in my mind that these symptoms of this breaking virus affected human behavior too, indirectly, that they set off the famous tulip craze in the Netherlands. Let me explain. In the 17th century, the Netherlands was among the most important trading centers in all of Europe, with lots of rich merchants who wanted to showcase their wealth, for example, by displaying exotic tulips in their private gardens. Now, tulips are not native to the Netherlands. They originated in the mountains of Central Asia and spread from Persia, present-day Iran, to the Turkish Ottoman Empire, and from there eventually reached Europe. There's an explanation for the origin of the name tulip that kind of reflects this. Apparently, it came from a Persian word for turban. You know, a cloth wound around the head, a style of headgear worn by men in that part of the world. The Ottomans used a similar name for tulips after they acquired them from Persia. And then, in the late 16th century, a variation on that name found its way from what is now Turkey to the Netherlands, along with the plant itself. Okay, a tulip, of course, is typically grown from an underground bulb. And although, as it flowers, the old bulb shrivels to almost nothing, the plant produces another large bulb, and maybe 2, 3, 4 smaller ones to take its place. And while tulips take up to 7 years to bloom if you start from seeds, a large bulb can produce a flower the very next year. With the smaller bulbs, it takes maybe a couple of years. And since these bulbs remain viable for quite a long time, even out of the ground, they can be stored or transported long distances without much of a problem, which helps explain the spread of tulip cultivation. At first, tulips were rare in the Netherlands and only for the wealthy. But in the early 17th century, as more bulbs were produced there, you'd think the prices would come down. In fact, though, the popularity of some tulips increased tremendously, so demand soon far exceeded supply, and their prices skyrocketed. And the tulips most prized for their uniqueness and beauty were apparently the ones infected by this mysterious virus. At the time, nobody was really able to breed tulips like these. Color breaking happened in just 2 or 3 out of 100 bulbs, and seemingly just by chance. And since you didn't really know when you bought a bulb if the colors would break, well, Dutch speculators invested hand over fist and drove prices sky high. Some bulbs, even while still in the ground, were sold for as much as you would pay for a house at the time. But the huge speculative bubble created by this tulip craze eventually collapsed. And when the prices fell, that wiped out a lot of fortunes almost overnight. Later on, tulip breeders learned to duplicate color breaking in healthy, uninfected plants. So the spectacular-looking tulips so common today are the result not of chance viral infections, but of carefully controlled breeding.
What does the professor mainly discuss?
Listen to part of a lecture in a botany class.
When we talk about pollination ecology, we're talking about the relationship between a plant and its pollinator. From the plant's perspective, the ideal pollinator is an animal that is underfed, ready to eat, and in a hurry. The pollinator, on the other hand, wants to remain well-fed with as little effort as possible. These factors help drive the evolution of plants and their pollinators, both of which depend on this balanced and delicate relationship. Sometimes only certain insects or birds can pollinate certain plant species. So, to really understand pollination ecology, both the flower and its pollinators must be studied. Let's start with flowers. There are several important factors associated with pollination: when and how often a plant flowers, how long the flowering cycle lasts, and the number of flowers that open at the same time. For example, flowering may coincide with the migration of a certain animal species that pollinates the plant, or producing many flowers at once may increase the number of pollinators a plant attracts. Other characteristics of flowers are also important. Features such as color, scent, and shape attract pollinators, as does the reward in the flower, the pollen or the nectar, that feeds the pollinator. For example, flowers that attract bats tend to be green or cream-colored because visibility isn't important. Bats are practically blind, remember, and these flowers bloom at night when bats are active. Now, there's a flower in the Amazon rainforest called a royal water lily, and the characteristics of its flowers change during the pollination process. The royal water lily uses color, temperature, and scent to attract the beetles that pollinate it. When the flowers of the royal water lily first open up, when they first bloom, they're white. They also emit a strong odor, and their temperature rises. Producing heat serves two purposes. It magnifies the scent of the flower, and it helps the beetles maintain their body temperature. When a beetle arrives at the flower closes around it for about 24 hours, so that the beetle becomes covered with pollen. Then, when the flower opens, its color changes to red, and it cools down. When the beetle flies out, it carries the pollen to a different heated, white, fragrant flower. As you can see, plants go to a lot of trouble to attract attention. So, what kind of attention are they attracting, and why? Well, sometimes flowers provide shelter for insects, a place to lay eggs, for instance. But usually the attraction is food, nectar and pollen. Nectar is mainly a sugar solution, while pollen is a grain made up of part of the plant cell structure. In both nectar and pollen production, quality and quantity vary over time, but they're always related to the needs of the pollinator. You can see that the relationships between pollinators and plants are delicate, so any number of factors can disturb them. Human development is one, and agriculture is generally believed to be the most harmful. It can fragment habitats in a variety of ways, reducing the number of pollinators, which in turn may reduce the number or size of the flowers, which of course affects the animals that feed on them. Exotic plant species not native to the area can move in and compete. Even bees brought in to pollinate crops can alter natural pollen dispersal systems of rainforest plants. On the other hand, recent studies have shown that the disruption of one aspect of the pollination cycle doesn't usually lead to the extinction of other species. It turns out that plant-pollinator relationships are more adaptable to change than we thought. So really, it's hard to know just how agriculture affects the pollination of plants.
What is the lecture mainly about? Listen to part of a lecture in a botany class.
Okay, last time we talked about photosynthesis, the process by which plants use light to convert carbon dioxide and water into food. Today, I want to talk about another way light affects plants. I'm sure you all know from physics class about how light moves in microscopic waves, and that we can only see light when the wavelength of that light is in a specific range. Plus, depending on the wavelength, we see different colors. Well, plants are also capable of distinguishing between different wavelengths of light. Now, I don't want to confuse you. It's not like plants have eyes. Plants don't see in the sense that humans or animals do. But they do have photoreceptors. Photoreceptors are cells that respond to light by sending out a chemical signal, and the organism, the plant, reacts to this signal. In fact, the signals that plants get from their photoreceptors sometimes cause significant reactions. And many plants are seasonal, and one way they know when winter is ending and spring is beginning is by sensing the change in light. The time when an adult plant flowers is based on the amount of light the plant senses. Certain plant species won't flower if they sense too much light, and some plants will only flower if they sense a specific amount of light. Of course, these aren't conscious reactions. These plants just automatically respond to light in certain ways. Plants are also able to distinguish between specific wavelengths of light that the human eye cannot even see. Specifically, there's a wavelength called far red. Although, why they call it far red? I mean, it's not really red at all. It lies in the infrared range of the spectrum. We can't see it, but plants can sense it as a different wavelength. Okay, now I need to mention another thing about photosynthesis. I didn't explain how different wavelengths of light affect photosynthesis. When a plant absorbs light for performing photosynthesis, it only absorbs some wavelengths of light and reflects others. Plants absorb most of the red light that hits them, but plants only absorb some of the far red light that hits them. They reflect the rest. Remember this, because it's going to be relevant in an experiment I want to discuss. This fascinating experiment showed that plants not only detect and react to specific wavelengths of light, plants can also detect and react to changes in the ratio of one wavelength to another. This experiment was called the Pampas experiment. The idea behind the Pampas experiment had to do with the response of plants to changes in the ratio of red light to far red light that the plants sensed with their photoreceptors. Some biologists hypothesized that a plant will stop growing if it's in the shade of another plant, a reaction that's triggered when it senses an unusual ratio of red light to far red light. Okay, imagine there are two plants, one below the other. The plant on top would absorb most of the red light for photosynthesis, but reflect most of the far red light. That would lead to the plant in its shade sensing an unusual ratio. There would be less red light and more far red light than normal. What that ratio signifies is important. A ratio of less red light to more far red light would cause a reaction from the plant. It would stop growing taller, because that plant could sense it wasn't going to get enough sunlight to provide the energy to grow large. To test their hypothesis, researchers took some electrical lights. Actually, they were light-emitting diodes, or LEDs. These light-emitting diodes could simulate red light. So they put these LEDs around some plants that were in the shade. The LEDs produced light that the plants sensed as red, but, unlike sunlight, the light from these LEDs did not support photosynthesis. So the plants sensed a proper ratio of red light to far red light and reacted by continuing to grow taller, while in reality these plants were not getting enough energy from photosynthesis to support all of that growth. And because they weren't getting enough energy to support their growth, most of the shaded plants died after a short time.
What is the lecture mainly about?
Listen to part of a lecture in a marine biology class.
Okay, today we're going to continue our discussion of plant life in coastal salt marshes of North America. Salt marshes are among the least inviting environments for plants. The water is salty, there is little shade, and the ocean tide comes in and out, constantly flooding the marsh. So the variety of plants found in salt marshes is limited, but there is a plant genus that thrives there, the Spartina. In fact, the Spartina genus is the dominant plant found in salt marshes. You can find one type of Spartina, salt marsh cordgrass, growing in low marsh areas. In higher marsh areas, you're likely to find a Spartina commonly called salt meadow hay. So, how is the Spartina able to survive in an environment that would kill most plants? Well, it's because salt marsh grasses have found ways to adapt to the conditions there. First of all, they're able to withstand highly saline conditions. Now, one really interesting adaptation is the ability to reverse the process of osmosis. Typically, the process of osmosis works, well, when water moves through the wall of a plant's cell, it will move from the side containing water with the lowest amount of salt into the side containing the highest amount of salt. So imagine what would happen if a typical plant suddenly found itself in salt water. The water contained in the plant's cells, that's water with very little salt, would be drawn out toward the seawater, water with a lot of salt. So you can see the fresh water contained in the plant would be removed, and the plant would quickly lose all its water and dehydrate. But what about the Spartinas? Well, they allow a certain amount of salt to enter their cells, bringing the salt content of the water within the plant to a slightly higher concentration than that of the surrounding seawater. So instead of fresh water moving out of the plant's cells, salt from the seawater enters, reverse osmosis, and this actually strengthens the cells. Another adaptation to the salty environment is the ability to excrete excess salt back into the environment. That's why you might see a Spartina shimmering in the sunlight. What's reflecting the light is not salt from seawater that has evaporated, although that's a good guess, but it's actually the salt that came from within the plant. Pretty cool, huh? You can really impress your friends and family with that little tidbit the next time you're in a salt marsh. But coping with salt is not the only challenge for plants in a salt marsh. Soil there is dense and very low in oxygen. So Spartinas have air tubes. Air enters through tiny openings on the leaves. The tubes provide a direct pipeline for the oxygen, carrying it down the leaves through the stems and into the roots, where it's needed. If you pull up a Spartina, you might even notice some reddish mud on some of the roots. This is caused by oxygen reacting with iron sulfide in the soil, and it produces iron oxide, or rust. Now, although the Spartinas have adapted several chemical and physical mechanisms that allow them to thrive in salt water and to feed oxygen to their roots, there's yet another aspect of the harsh environment that they've had to adapt to, the force of the tides and occasional violent storms. Wind and water are constantly crashing into these plants. So, as you might have guessed, they've developed a means of solidly anchoring themselves into the soil. How? Well, they have tough sort of underground stems called rhizomes. Rhizomes from one plant grow through the muddy soil and interlock with those of other nearby plants. The plants form a kind of colony, a community that will survive or perish together, because alone, as single plants, they cannot survive. Of course, the plants in these colonies also need tough, resilient stems above the soil, stems that can bend a lot but not break as water constantly crashes into them. So, in addition to the interlocking underground rhizomes, they have yet another adaptation, and it's, well, we're back to reverse osmosis again. By adjusting the osmotic pressure so that its cells are always fully inflated, the plant is able to withstand great pressure before snapping. So Spartinas may look like simple marsh grass, but they're really a wonder of chemistry, physics, and structural engineering that allows them to survive and even thrive in an environment in which most other plants would wilt and die within hours.
What is the lecture mainly about? Listen to part of a lecture.
Listen to part of a lecture from a botany class.
Hi, everyone.
Good to see you all today. Actually, I expected the population to be a lot lower today. It typically runs between 50 and 60% on the day the research paper is due. I was hoping to have your exams back today, but the situation was that I went away for the weekend and I was supposed to get in yesterday at 5, and I expected to fully complete all the exams by midnight or so, which is the time that I usually go to bed, but my flight was delayed and I ended up not getting in until 1 o'clock in the morning. Anyway, I'll do my best to have them finished by the next time we meet. Okay, in the last class we started talking about useful plant fibers. In particular, we talked about cotton fibers, which we said were very useful not only in the textile industry, but also in the chemical industry and in the production of many products, such as plastics, paper, explosives, and so on. Today we'll continue talking about useful fibers, and we'll begin with a fiber that's commonly known as manila hemp. Now, for some strange reason, many people believe that manila hemp is a hemp plant, but manila hemp is not really hemp. It's actually a member of the banana family. It even bears little banana-shaped fruits. The manila part of the name makes sense because manila hemp is produced chiefly in the Philippine Islands, and of course, the capital city of the Philippines is Manila. Now, as fibers go, manila hemp fibers are very long. They can easily be several feet in length. And they're also very strong, very flexible. They have one more characteristic that's very important, and that is that they are exceptionally resistant to salt water. And this combination of characteristics, long, strong, flexible, resistant to salt water, makes manila hemp a great material for ropes, especially for ropes that are going to be used on ocean-going ships. In fact, by the early 1940s, even though steel cables were available, most ships in the United States Navy were not moored with steel cables. They were moored with manila hemp ropes. Now, why was that? Well, the main reason was that steel cables degrade very, very quickly in contact with salt water. If you've ever been to San Francisco, you know that the Golden Gate Bridge is red. And it's red because of the zinc paint that goes on those stainless steel cables. That if they start at one end of the bridge and they work to the other end, by the time they finish, it's already time to go back and start painting the beginning of the bridge again, because the bridge was built with steel cables, and steel cables can't take the salt air unless they're treated repeatedly with a zinc-based paint. On the other hand, plant products like manila hemp, you can drag through the ocean for weeks on end. If you want to tie your anchor to it and drop it right into the ocean, that's no problem, because plant fibers can stand up for months, even years, in direct contact with salt water. Okay, so how do you take plant fibers that individually you could break with your hands and turn them into a rope that's strong enough to moor a ship that weighs thousands of tons? Well, what you do is extract these long fibers from the manila hemp plant, and then you take several of these fibers and you group them into a bundle, because by grouping the fibers you greatly increase their breaking strength. That bundle of fibers is much stronger than any of the individual fibers that compose it. And then you take that bundle of fibers and you twist it a little bit, because by twisting it, you increase its breaking strength even more. And then you take several of these little bundles and you group and twist them into bigger bundles, which you then group and twist into even bigger bundles, and so on, until eventually you end up with a very, very strong rope.
What aspect of manila hemp fibers does the professor mainly discuss in the lecture?